Our introduction to the nervous system is going to focus on the functional units of the nervous system, which are the neurons and the glial cells. So before that, we want to understand how the nervous system operates. And in essence, we're taking in sensory information, integrating or processing that information, and then producing a response. So think of it as simply cause. Notice also that our stimuli, our changes, are inside and outside. So we have both internal and external monitoring that takes place. We have to have an interpretation of that information, and then we have to produce a response. The other thing to notice is this word right here, motor. The motor response is in almost always, a muscle or a gland that produces the response. So, the neuron. Think of the neuron as the functional unit of the nervous system. All of the information being brought to, so the sensory information, the ability to process the information, and the ability to send out the information, the response, is all carried by neurons. Now, when we talk neuron anatomy, there's two basic parts. You have the cell body and you have the extensions of the cell body. So the cell body has specialized structures associated with the function of the nervous system, so the ability to respond to some form of an action potential, in other words, something that disrupts the resting membrane potential. And then we have extensions capable of carrying that information to and from the cell. So the main thing here is to pick up on the very different components of the cell compared to something like the muscle cell. So it goes back to that idea of form and function. Now the extensions are actually kind of important, and we'll talk about the classifications. We have structural classifications of neurons associated with these extensions. But the main thing to recognize is notice the arrows. The information comes in and the information goes out. In other words, when we talk about the neuron, all the information is carried in a single direction, to the cell body and from the cell body via these extensions. Now, the other thing to pick up on are these Schwann cells. In other words, the Schwann cells are basically fat. And the Schwann cells, which are in essence a glial cell, play a huge role in the actual action potential. Remember in the video it talked about how signals can be transmitted at tremendous speeds from your brain to your foot or something like that? Well, part of the ability to be fast is the presence of these, which creates this, what's known as the nodes of Neuron VA, and produces what's known as saltatory conduction. We'll talk about that when we talk about the action potential. Now, just before we get into the action potential, there's a few things you have to remember. Notice that our neurons are irritable, meaning they respond to stimuli, and they're conductive, meaning we have a resting membrane potential. We have those voltage-gated channels that control what ions enter and leave the cell. And remember that this resting membrane potential is the result of the sodium-potassium pump, where we put 3 sodium ions outside compared to 2 potassium ions that we put inside. And so it is this electrochemical gradient that actually allows an action potential, or the depolarization of the membrane, which causes the action potential to travel. And the depolarization is simply that flow of sodium in, and at a certain point we see the flow of potassium out, and then we see that repolarization wave follow, where we reset the entire mechanism. Now, the resetting of the mechanism, because it is a sodium-potassium pump, requires ATP. So remember, the ATP actually causes the configuration of the protein. Changing the shape of the protein changes the function, and so we see that sodium-potassium exchange. Now, let's go back and talk our saltatory conduction here. When we look at the action potential, notice that we have the information come in. Notice the changes to the sodium-potassium. Saltatory conduction, basically what it does is, since it's fat or it's lipids, ions and fats don't mix. So what happens is I actually cover up a bunch of this area with fat, and the action potential will literally jump from node to node. So when we look at this, let's go back to our cell here, what literally happens is these openings, which are the nodes of Neuron VA, the action potential literally jumps. So what I see is, I see something that's going to be done much faster, because technically the actual space involved is dramatically cut in half. And so this is why the speeds of conduction are so much greater. The other thing to keep in mind is this is actually also where we get the difference between white and gray matter. The presence of the fat, the myelin, is what creates the white matter in the central nervous system. Now, neuron classification is actually by two different ways. We have what's known as the functional classification, which is sensory, motor, and interneurons. Notice their location. So I have a sensory motor that has the afferent transmission. So that sensory is basically my cause. It goes through an interneuron, which is within the central nervous system, and I produce my effect via the efferent transmission. So pay attention to the terminology. What's important to note as well is that there are dedicated pathways. I have an afferent pathway, I have an efferent pathway. And these dedicated pathways are important when we look at how the nervous system is able to do what it does. So we have information in, processed, information out. This is also the basis of the reflex arc. Reflexes you're already fairly familiar with. You touch something hot or sharp, your finger immediately moves. And what's kind of cool is that your finger will actually move before you're consciously aware of why it moved. And it's just a fractional difference, but do not try this at home. I'm not telling you to go put your hand on the stove and see if that's the case, but you'd be surprised once you hear this how the next time you're kind of aware of, wait, I really was moving before I knew. And that's in essence the reflex arc, because sometimes in order to maintain homeostasis, in order to survive, you have to be able to immediately respond without necessarily the brain without you consciously processing information. The spinal cord is a component of the central nervous system. So integration can occur in the spinal cord, and that's what's going on with the interneuron within this reflex arc. Okay? Now, the other way neurons are classified is structurally. So notice now what's going on with those extensions from the cell body. This is the multipolars, what we typically think of as a cell, as a neuron. This is the most commonly associated with our effector neurons. So the efferent neurons are very much multipolar neurons. But then we see other things. We don't necessarily need a neuron quite like this in the brain. We don't necessarily need a neuron like this in terms of our sensory neuron. And so that's where you see these different classifications. And the classification always revolves around the extensions from the cell body. So notice here, the cell body's out of the way, and that's the difference between the unipolar and the bipolar. The bipolar is within the path. Now, the majority of our neuron cell bodies are within the central nervous system, and this is actually part of why the central nervous system is so well protected. Surrounding the spinal cord and surrounding the brain, we have very intensive bony structures. We don't have bony structures surrounding the neurons, surrounding the sensory and efferent neurons. And so you can see that our cell bodies are here within the central nervous system. But occasionally we have cell bodies that aren't, and that's what's going on here, and that's what we term a ganglion. Now, the cell bodies are typically what we associate with gray matter because they're not going to be enclosed by myelin sheaths. The myelin sheaths are what produce the white matter. But within our central nervous system, we have both gray and white matter, so keep that in mind. So our action potential involves stimulation at the dendrites. We see the action potential propagate down the axon to what's known as the axonal knob. Well, great. Notice there's no actual touching. Remember, within the neuromuscular junction, there was no actual touching of the neuron to the muscle at the neuromuscular junction. We see exactly the same thing within the nervous system. We have the formation of synapses, and it's this cleft, it's this gap between the neurons. And the reason being is we want the neurotransmitter to have to diffuse across. We need that time that gives us, and to control how the action potential is going to work, because we can actually inhibit or stimulate the neurons in terms of what happens when I add neurotransmitters. I can actually elevate the resting membrane potential so that it's easier for a neuron to fire. I can also lower that and inhibit the ability of the neuron to fire. And this is all done, in terms of what's going on here in the synapse and at the cleft. The other thing to remember is, just like when we talked about acetylcholine diffusing across, that's exactly what's going on between neuron cells. So we have this synapse, we see neurotransmitters diffusing across, They bind to a membrane protein. The membrane protein is associated with that voltage-gated channel for sodium and potassium, and we cause a change to the shape of the protein. We cause a change in the permeability of the membrane. And remember, ultimately, it's that changing permeability to sodium and potassium that produces our action potential, that produces the depolarization and repolarization that follows. So this is, in essence, it's very similar to the neuromuscular junction, except we just have different parts. We have the axonal knob, we have the dendrite of the adjacent neuron, and we see everything firing around. And neurotransmitters play an important role. We'll talk about specific neurotransmitters when we deal with the brain and the different nervous systems. Now, the supporting cells, which we call the neuroglial cells, they actually are more than just supporting cells. They are significant to the function of and the health of the nervous system as a whole. Now, astrocytes are huge for this reason right here. They are responsible for what's known as the blood-brain barrier. Without the blood-brain barrier, our brain literally stops functioning. We have specialized fluid called cerebral spinal fluid, and it's similar but just different enough from blood plasma that we maintain a completely different chemical environment within our central nervous system. Now, other cells play other roles. So you can see the microglia, they have kind of, since they dispose of debris, think of them as kind of like associated with the immune system, with the health of the overall environment. The ependymal cells are what produce the cerebral spinal fluid and circulate the cerebral spinal fluid. The oligodendrocytes are simply the cells that produce myelin sheaths within the central nervous system. So we have Schwann cells within the peripheral nervous system, or within the system we typically think of, and then we have oligodendrocytes within the central nervous system. The central nervous system being the brain and spinal cord, the peripheral nervous system is everything else. All right, so you kind of get an idea of the various glial cells and their functions.